Hi there, my name is Viraj Alimchandani and I'm a PhD student at the University of Montréal. I work at the Plant Biology Research Institute, which is based in the Montreal Botanical Garden, and I study the biophysical aspects of plant growth and anatomy using exciting microscopy and modelling techniques. I come from London, in England, so moving to Montreal was a big leap for me, especially as my French skills leave a lot to be desired. However, big moves are not strange to my family. All my grandparents grew up in northern India, and they all emigrated in the 1950s, shortly after the partition in 1947, and one set moved to Hong Kong and the other set moved to London. Learning and good academics were an important part of my upbringing, as my mum was a teacher, and at high school I really enjoyed maths and physics, which made me want to take things further at university. Sadly, the biology curriculum at my school wasn't terribly interested, so I think if you told 16-year-old me that I'd be doing a PhD in biological sciences, I'd be very surprised. I began as a physics undergraduate at the University of Oxford, where I was enthralled by the real-world applications of maths and physics, especially in relation to fluid dynamics, climate dynamics, and nanoscale mechanics. The combination of these interests led me to undertake a biophysics mini-project during my bachelor's, where I delved deep into optical microscopy and the maths behind how it actually works. I even got the chance to have free rein over the samples I investigated. I then went on to explore different types of microscopy and discovered atomic force microscopy, or AFM. This form of microscopy is essentially a tiny probe which is driven by an electrical signal which makes physical contact with a sample and can uncover a sample's intrinsic mechanical properties. In essence, this tiny probe pokes the surface of a sample, and its feedback is converted into a digital signal which can tell us about the sample's mechanical properties. This high-precision real-world application of physics really played to my interests, so I sought out a master's project which would allow me to learn more about these techniques. The research group I joined for my master's project was amazing, and my supervisor and the fellow PhD students really sparked my profound interest for biology. Not only was I able to learn about AFM in greater detail, but I was exposed to a whole slew of different microscopy and analysis techniques which I used across a wide variety of interdisciplinary studies, such as magnetic force microscopy, or MFM, scanning electron microscopy, or SEM, gel imaging, and fluorescence imaging. I used these imaging techniques in my project which paired materials science with biology, and it really got me interested in life at the nano and micro scale and the interaction between biological molecules and lab-synthesized materials. One of my long-term goals is to use all these techniques to aid the crises facing the ecology and the environment in the future. This was also my first experience in a proper research laboratory, and I was able to collaborate with people from all sorts of different backgrounds, from maths and physics to biology and medicine. That year really showed me the power of interdisciplinary study. Research areas today are so diverse that no one can be an expert in everything, so it's important to have lots of people with expertise from complementary backgrounds. There were also several other projects happening in my master's lab, one of which was investigating plant growth at the nanoscale using AFM. And although I wasn't directly involved, I found it fascinating how you could probe growth at the nanoscale without destroying any samples. All of this led me to apply for a PhD looking at plant biophysics from a microscale perspective. My current research is just that, and it also relies heavily on microscopy techniques, although this time they are confocal microscopy and cellular force microscopy, or CFM. Combining these with some modelling techniques, I analyse how physical forces affect cell growth in plants. More specifically, I use confocal microscopy to image samples, which can be put into a modelling software to track cell growth and lineage. This can tell us how the cells in a plant organ are growing and how this growth changes over time. I also use CFM to investigate the mechanical properties of cells. Much like AFM that I described earlier, CFM also uses a probe to actually poke the plant cells to see how they respond, and this can give us information about the cell's mechanical properties, such as the cell wall stiffness. As this research is right on the cusp of biology and physics, the lab is full of people from different backgrounds, which makes discussion and collaboration that much more exciting because you're learning new things every day. Although it's quite technical, my research has a very real relevance to ecology and evolution. Investigating the physical and mechanical properties of cells can lead to understanding how these microscale factors can control the resilience of plants against environmental stresses such as high winds or heavy snow. This knowledge can be extremely useful in many fields, both applied and fundamental. In agriculture, it could serve as a basis to develop new crops which could withstand high winds or heavy snowfalls without being destroyed. In ecology, my research will help understand why some species are better adapted to harsh physical conditions than others. Finally, linking cell biophysics to genetics will help us understand how this physical resilience has evolved in some plants. An important point to remember is that one person's research alone will not be able to achieve this. It will be the collaborations with other scientists with different expertise which will be able to tie all these different strands of research together. The main thing I've learned during my time in research is that collaboration is key for interdisciplinary study, which in turn is key for the advancement of science.